Welcome. Here, in this module, we'll look at some details of the second stage of glycolysis, including a redox reaction involving the transfer of electron pairs to electron carriers, a few more regulated enzymes, and the synthesis of ATP in coupled reactions. As we go through the reactions, remember that each is occurring twice per starting molecule of glucose. In the first reaction of the second stage, glyceraldehyde 3-phosphate is oxidized and simultaneously phosphorylated to make a molecule of 1,3-diphosphoglyceric acid. The oxidizing agent is NAD+, which is reduced by electrons in a hydride ion to NADH. NAD+ then is an electron carrier. The enzyme for this reaction is glyceraldehyde 3-phosphate dehydrogenase. You'll see that most biological redox reactions are in fact catalyzed by dehydrogenases, meaning removal of hydrogen, because they usually involve the movement of electron pairs on either hydride ions or complete hydrogen or H2 molecules. This reaction is yet another endergonic reaction based on its energetics in a closed system under standard conditions, where delta G0 is equal to plus 1.5 kilocalories per mole. The reaction is also reversible, and it's also allosterically regulated, though in an unusual way that I'll show you in just a moment. I want to remind you about the electron carrier NADH and a few others. NADH is nicotinamide adenine dinucleotide, and it's an electron carrier, as are its phosphorylated form, NADPH, as well as FADH2. In their oxidized forms, NAD+, NADP+, and FAD, they accept electron pairs from reduced molecules, again, either as a hydride ion or as whole hydrogen molecules. As reduced electron carriers, NADH and FADH2 have captured a significant amount of the free energy that once was in a glucose molecule. NADPH, we'll see later, serves as an oxidizing agent, capturing electron pairs and free energy during photosynthesis and in other cellular redox reactions. Before I describe the unusual allosteric regulation of glyceraldehyde 3-phosphate dehydrogenase, let's consider this question. What would a high concentration of NAD+ mean to a cell? First, it means that cells are actively respiring. It means that the cell is rapidly oxidizing NADH, and that's what's keeping NAD+ levels relatively high. You may recall that NADH oxidation fuels ATP synthesis. This happens in electron transport and oxidative phosphorylation. So high NAD+ levels must mean that the cell is actually calling for more ATP to be synthesized. So how should high NAD+ levels affect the G3P dehydrogenase reaction? Clearly, it should make the enzyme catalyze its reaction faster, so that it will not be the rate-limiting reaction in the glycolytic pathway, meaning it won't be the slowest reaction in the pathway. So here's how it works. Glyceraldehyde 3-phosphate dehydrogenase is made up of 4 identical polypeptide subunits, each with an active site able to catalyze the redox reaction. Regulation, or allosteric regulation, is by negative cooperativity. Let's have a closer look. After one subunit binds NAD+, the remaining subunits have a lower affinity for NAD+. That means that more NAD+ needs to be around before a second subunit will bind a molecule of NAD+. So if the concentration of NAD is high enough, another NAD+ can bind to a second subunit. But this in turn will lower the affinity of the 2 remaining subunits for NAD+, and so on and so on. What that means is it'll take very high levels of NAD+ to bind all 4 subunits of glyceraldehyde 3-phosphate dehydrogenase, so that the enzyme will be most active, that is to say, it will reach its Vmax or maximal rate of catalysis, only when NAD+ levels are at very high or relatively very high concentrations. In the next coupled reaction, a phosphate is transferred from 1,3-diphosphoglyceric acid to ADP to make ATP by substrate level phosphorylation, leaving behind 3-phosphoglyceric acid. Now remember again, this reaction, like all of those in the second stage of glycolysis, is occurring twice per starting molecule of glucose. The reaction is catalyzed by phosphoglycerate kinase. It's also exergonic with a standard free energy change at delta G0 of minus 4.5 kilocalories per mole. In this reaction, The endergonic synthesis of ATP by substrate-level phosphorylation is coupled to a much more exergonic reaction, the hydrolysis of 1,3-diphosphoglyceric acid. This reaction is reversible, the arrows can tell you that, and in active cells it's very rapid, which, by the way, is why we saw earlier that the concentration of G3P in cells is usually very low. Now, why do you think we would refer to 1,3-diphosphoglyceric acid as having a very high energy phosphate linkage? Because the hydrolysis of a phosphate from 1,3-diphosphoglyceric acid is very exergonic. To demonstrate this for yourself, write the reaction as the sum of an ATP dehydration synthesis reaction plus a 1,3-diphosphoglyceric acid hydrolysis reaction. You can refer to some of the examples of this sort of thing that I presented earlier to get you started. The next two reactions are each modestly endergonic and freely reversible. First, the phosphate group on the 3-phosphoglyceric acid, or 3PG, is shifted to the number 2 carbon by a mutase in a reversible endergonic reaction. Then, 2PG is converted to phosphoenol pyruvate, or PEP, in another reversible endergonic reaction. Like 1,3-diphosphoglycerate, phosphoenol pyruvate, PEP, is also a very high energy phosphate compound. In this quite exergonic reaction, delta G0 equals minus 7.2 kilocalories per mole, the hydrolysis of phosphoenol pyruvate is coupled to the synthesis of another molecule of ATP. The enzyme catalyzing this reaction is pyruvate kinase. In energetic terms, the large amount of free energy released by the hydrolysis of the phosphoenol linkage in PEP is what drives the endergonic ATP synthesis. Again, demonstrate this to yourself by writing the overall reaction as the sum of two separate reactions. Let's look at the enzymatics, what I call the enzymatics. In this case, pyruvate kinase catalyzes a biologically irreversible reaction, and the enzyme is allosterically regulated. The enzyme is inhibited by ATP, citric acid or citrate, and long-chain fatty acids of the sort you would find after they were immediately hydrolyzed from the glycerol backbone. And the enzyme is stimulated by fructose 1,6-diphosphate and by phosphoenol pyruvate itself. What is the logic for how each of the allosteric effectors I just told you about regulates pyruvate kinase? Here's a balance sheet for glycolysis of a mole of glucose to 2 moles of pyruvate. We have consumed 2 molecules of ATP, but we synthesized 4. That's a net of 2 ATPs produced. The 2 ATPs account for a capture of approximately 15 kilocalories of the free energy once in glucose. We end up with 2 molecules of pyruvate, as well as 2 molecules of NADH, along with 2 protons. Well, where did the protons come from? In glyceraldehyde 3-phosphate oxidation, a whole hydrogen molecule is removed from G3P, but NAD+ grabs only the hydride ion. That leaves a proton behind in solution, and that's where the proton comes from. The standard free energy change for the whole of glycolysis through pyruvate can be summed up. Just add all the free energy changes for the reactions as I've shown them to you, and it comes to minus 44 kilocalories per mole of glucose. Now that's 44 kilocalories out of a potential of 686 kilocalories for the whole respiratory pathway from glucose and oxygen to water and CO2. So at this point, where is the free energy that was once in a molecule of glucose or in a mole of glucose? Well, a small amount is in ATP, so most of the rest must still be in the 2 pyruvates and in the 2 NADH molecules. Let's take a moment to look at some of the different phosphate linkages, some of which release a lot of free energy when hydrolyzed, and some of which do not. First off, the hydrolysis of an enol phosphate linkage, such as in phosphoenol pyruvate, and the phosphoanhydride linkage in 1,3-diphosphoglycerate are both highly exergonic. Hydrolysis of the phosphate linkage in creatine phosphate, shown here, is also very exergonic. You can look up the role of creatine phosphate in providing energy to make ATP for muscle contraction on your own. Hydrolysis of the ATP phosphoanhydride linkage is also exergonic, but not as much as the linkages in PEP or 1,3-dipG. So calling ATP a high energy intermediate was in fact a good call. Lastly, there are many phosphate ester linked molecules, We've seen some of them, like glucose 6-phosphate shown here, or AMP, adenosine monophosphate, whose hydrolysis, while exergonic, will not release enough free energy to be coupled to ATP synthesis. Glycolysis from glucose to 2-pyruvates is sometimes called incomplete glycolysis. Complete glycolysis is a fermentation which occurs under anaerobic conditions. Let's look at one that you may already be familiar with, the reduction of pyruvate to lactate or lactic acid. This exergonic reaction is catalyzed by the enzyme lactate dehydrogenase, or LDH. Catalysis by a dehydrogenase gives away that this is a redox reaction. The enzyme is not allosterically regulated, but it actually comes in different shapes, some of which are reversible and others less so, or in reality, never reversible. Let's look at this enzyme a bit further. LDH is actually a tetramer made of two different monomer polypeptides called M, as in Mary, and H. In theory, up to 5 different LDH tetramers could be assembled with these two monomers, as shown here. You could have 4 M's, or 3 M's and 1 H, or 2 M's and 2 H's, or 1 M and 3 H's, or just 4 H's. In real life, some types of cells will have more H monomers in the LDH molecules, others more M monomers. In other words, there's an equilibrium in the concentration of all possible tetramers that tends towards M-rich or H-rich, or possibly equal amounts of all the tetramers, depending on the different proportions of the two monomers synthesized in the different cell types. Different versions of the same enzyme are called isoenzymes or isozymes for short. LDH can be extracted from different tissues, and the different isozymes separated by starch gel electrophoresis. So we're going to be separating the different isozymes pretty much by charge alone. Here are extracts of heart and skeletal muscle LDH. The samples are put into the wells of an electrophoresis gel, and the power turned on. At the end of electrophoresis, the gel is stained, in this case specifically for LDH. You can see that the different LDH isozymes are differentially charged in solution, and what you should see is that there are actually 5 different spots, each representing one of the different isozymes. It turns out that the M-rich, especially M4 LDH, is the least reversible. In fact, as I suggested a moment ago, in real terms it's never reversed, while H4 is readily reversible. Here's an explanation of how the LDH isozymes in different cell types serves our physiology. During strenuous activity, respiration cannot keep up with the demand for ATP needed to fuel skeletal muscle contraction. As a result, pyruvate builds up, and an M-rich LDH isozyme, in turn, reduces the pyruvate to lactate. The resulting buildup of lactic acid cannot be indefinitely sustained, causing the muscle ache you probably know as muscle fatigue. When you're finally forced to rest, lactic acid cannot be reoxidized to pyruvate because the M4 or M-rich LDH isozymes do not readily reverse the reaction, so the lactate is released into the circulation to be picked up by cells with more H-rich LDH isozymes. These cells oxidize the lactate and then the pyruvate for their own use. Heart muscle contains mostly H-based LDH isozymes, which makes sense since cardiac muscle relies exclusively on respiration for its ATP needs for contraction. And this concludes our presentations on glycolysis.